out of 7, okay? Be lots of pictures of things that you'll have boxes that are pointing to structures, okay? Be a lot of that. So that's the format. That'll also be true for exam 3. Now, with regard to exam 1, I know some of you are kind of fretting about the score. Remember, this is 15% out of 100%. So that would be, if you put it in points, that would be 15 points out of 100 points target for the whole course. So the difference between getting 50% on the first exam and 9% was 6 points. Okay. So just one exam said that it's very tough. You know, there was biophysics in there. There was electrochemistry that might not be so familiar with. And although we've held the exams now, had a discussion, we're going to hand the exams back later in the course together with exam 2, so you can have that for studying. But again, for your personal use. Okay, so that's been a bit delayed, but we have to organize to do that. Okay, so we'll be able to use that for review as well. So again, each of these exams, section exams, not really midterms, right? They are for that section. So this one will be for this section, and exam 3 will be for its section, and then the final will be cumulative. Okay? I think we are online now, so let's. Now, I posted a study guide. I'm going to turn this off. I hate doing this, but this is very dim. I posted a study guide I've had for a long time for neuroanatomy, and then because I've done all the developmental neuro this time, I made a study guide kind of for myself too. Okay, so that's actually on Lalema. You may or may not have seen it. Okay, so there is a study guide for the developmental part. And what I thought I would do is start out by going through the study guide, because it's just a nice way of just going through all the points, okay? And if you have a question, all right, or you just say, can we see more about that, we can go back to the original PowerPoint. Or I can just tell you, okay? Now, before we start, is anybody dying to ask a question? And you all know this is review, right? That we switched the lectures, if you're wondering, like, what's going on today? Okay, yeah, we switched the lectures. Nope, so let's dive in. Okay, what I thought I'd do is going to go through this stuff fairly efficiently, okay, not necessarily quickly, efficiently. Taking your questions or not. Well, I'll take the questions, but whether or not you have them, okay? And then try to get done early enough, and then stick around if those of you that want to ask questions outside the big group, okay? We'll see how that goes, but hopefully we'll have at least 20 minutes for that. So I don't want to race through this, and I think I should probably blow this up to much more readable. I'll see that in the back. You should have it. Yeah. So you found these study guides, yeah? Okay. So starting with the basics, all right. I'm going to have to stand here to the side. Starting with the basics, you really should know, the essential elements of the central nervous system, being the spinal cord, brain, and brainstem and brain. Okay. So again, anything outside that, even if it's the autonomic nervous system, the fibers, okay, are peripheral nervous system. So the autonomic nervous system, all the cell bodies, okay, for one part of the that nervous system, okay, or at least the main pathway, start in the central nervous system. And things like motor neurons, they are in the central nervous system. So it's the fibers that go out, okay. Now, we didn't really break down the peripheral nervous system because it turns out for the second stage peripheral nervous system in the autonomic side, there's actually ganglia outside, and that's listed here. Really want you to understand the concept. You don't need to know about autonomic ganglia, okay? So really just everything that's outside the central nervous system we say is PNS, okay? And then brainstem, medulla, pons, and midbrain, and this is distinct from the hindbrain. How? What is the hindbrain? Is it on there? It might be in there somewhere. If you find it, you can shout it out. You remember the difference with the hindbrain? So that's pons, medulla, and cerebellum. And you can say medulla. So that's the distinction. But brainstem really is fundamental because, in terms of function and integration, this all hangs together. Hindbrain is really a developmental thing. And obviously, the cerebellum is very different than the medulla and pons, despite the interaction. Okay, so the brainstem, the two branches of the autonomic nervous system, parasympathetic and sympathetic. The ventricles in the brain are, remember, there's the two lateral ventricles. They come together, so the fluid starts in the lateral ventricles, that is the cerebral spinal fluid, goes in, connects to the third ventricle, and then down to the fourth ventricle, okay? And then the next question is, where does it go? It flows out of the fourth ventricle. So it flows out of the fourth ventricle, and it communicates with what? Okay. It's chopped off. Nobody told me that. That's not good. Okay, this is a definite trick here to try to work on that. I think that's all. So it's running down over the spine, but what that really means is that it's not going through the middle of the spinal cord. It's going around the outside of the spinal cord. But where does it run within? That's within the meninges. Okay, so the covering, okay, and we'll come in those coverings in a second. Okay, is the outer limit, and the fluid's inside that. True in the spine and all the way up to the brain. And then where does the fluid go? I don't think I don't have this here, but could add in a little tidbit. So now so the fluid is made in the lateral ventricles in these specialized cells, and you make a lot of it all day, okay? And then it goes out, it flows, it goes out the fourth ventricle, goes over the brain, but also equilibrates to a degree with the fluid in around the spinal cord. And remember, we're erect, so a lot of that is like a gravity effect of mixing. But the retrieval of that fluid is where? That's at the top of the brain, out through the meninges. Okay, so eventually that fluid goes into the vasculature, goes into the venous return. Okay, so there always has to be an equation here. It's made on one side, has to be taken out from another side. Okay. So what are the meninges? The dura, the arachnoid, and pia mater. So I've left off dura, the mater here, the strong mother. Dura is okay. An arachnoid is basically the whole name. And pia would be okay. And so that's going outside to in. Remember, if you think about the dura, it sounds durable, right? That's the heavy outside part of the meninges, tough. All right, arachnoid's kind of spiral-like bit inside, and that's where the fluid is really running through. And then the pia is this thin little translucent layer that's over the surface of the brain. In fact, when you take people say they take the meninges off and look at the brain. So if the brain is removed, say an autopsy, and you look, it's very likely it still has pia on there. It would have to be peeled off before you'd look. And it does really look like some meat in a package. All right. Divisions of the spinal cord, 4. Cervical, thoracic, lumbar, and sacral. Okay. So with some of these, it's important to remember how many as much as to what they are. Okay. And this is top to bottom. Gray matter, those are the nerve cells. And the spinal cord that's inside, okay, what does it kind of look like? So you look at a cross section of the spinal cord, bottom to top, and you can see that gray matter inside. What does it have? It has these horns, right? So it looks kind of like a butterfly, or you could say it has horns. And they have horns that are in fact named, right? So the white matter is on the outside. The opposite is true in the brain, right? So now you get up into the brain. So it's true all the way through the brainstem, right? So this is really spinal cord and brainstem. Although the brainstem things get a little mixed up, so some of the tracks kind of move in, all right? So they're sort of outside, but they're getting a little jumbled, okay? Now you get all the way up into the brain. You get past the thalamus, you get to the cerebrum, the cerebral cortices, right? Gray matter on the outside, white matter collects on the inside. Remember the association fibers, the long fibers that collect to become the cortical spinal tract, and so forth. Sensory information coming up. So it sort of, inverts, right? And all the connections are now on the inside, and all the actions on the outside, if you will. Well, there's a lot of action on the white matter. Okay, So gray matter is divided into the dorsal horn and ventral horn. There was the answer for the question about what's the deal with the spinal cord. White matter and spinal cord has the ventral, lateral, and dorsal columns. Okay. So we have the dorsal columns are key, right? Because if we go to those now, they carry that sensory information. The basic sensory information are through the dorsal columns, all right? And then we have the spinal thalamic tract. And remember, that's in a funny spot. It's under the ventral horn of the gray matter. Okay. So this is a study sheet, and some of this, it's a lot easier to point to things. So we can go and review. In fact, maybe we'll just go ahead and do that. We'll just kind of cycle through some of the images. But it's good to have this so you can say what it is and then have it in your mind and then firm it up by seeing it. I'm visual. I like to see it first. But when you hear it, you have to remember it, you have to have that image in your mind, okay? So, and then there's the cortical spinal tracts bringing stuff down. Remember, those are the lateral tracts. And then we've already talked about the somatosensory system. And a key thing there is that there are, for both the somatosensory system and the spinal thalamic system, there are 3 synapses. That's starting from the point of origin. So whether it's some kind of proprioceptive, like a pressure sensor that's out in your skin, okay, versus pain, all right? Remember, those are free nerve endings, all right? They go through and on up, and they make 3 synapses, and the final synapse then is up in somatosensory cortex. All right. So the difference with a somatosensory system, that pressure touch, right, is what? So that information goes into the spinal cord, goes up the dorsal columns, and where does it stop? What does it say? Oh, it doesn't say. Remember? We can look at the picture in a minute. So with somatosensory one, so remember, somatosensory, that's the touch and pressure, okay? Sometimes called proprioceptive, okay? So it comes in, goes right into the dorsal columns, right? Stays on the same side, and then goes up and stops where? Medulla. Brainstem is fine, but medulla, right? Then what does it do? Then it crosses. Crosses goes over, and then it stops where? The thalamus. Remember, all sensory stuff has to go through the thalamus. Okay. Descending motor does not go through the thalamus, but all that motor integration goes back up. So that's a feedback loop, right? So that's true with spinal thalamic, right? And you know that why? Because it's in the name, right? So what happens with the spinal thalamic? Pain information comes in to the spinal cord, and in both cases, for the touch and the pain, where does it go? Generally, front or back? Comes in dorsally, right? So all sensation comes in dorsally through the dorsal horn, all right? And that makes sense, right? Because we just talked about the dorsal columns. Same for spinal thalamic. Comes in, makes a synapse though, right? In the dorsal horn of the gray matter. Then what does it do? That's the spine part. Crosses, goes up to the thalamus. Thalamus up to somatosensory cortex. Still 3. Okay, so 3, and then corticospinal, starting the cortex going down. If somebody just said corticospinal, you should think right off the bat we're talking about the motor system, right? Because now it's starting at the top and going down. If we're starting at the bottom and we're going up, that's got to be sensory. We bring in sensory information, we send down motor information. Right? Corticospinal, all right? 2 synapses all the way. This is what we call a final complement pathway, right? So after all that computation using the basal ganglia and some information from the cerebellum, and then there's like 3 or 4 other tracts and nuclei that have to do with the motor system, which you don't have to know. Okay. After all that computation, goes back to the primary motor cortex, and then there's a cell body there that sends an axon all the way down to the synapse. That's why it's cortical spinal, right? So that's one cell, one synapse, hits the motor neurons, goes out to the muscle, synapse number 2. So 3, and 2. 3 divisions of the brainstem, okay? And it would be good to know some basics about those functions, but particularly in the medulla, very basic, I hate to say primitive, but very basic, essential functions, right? Like the respiratory circuitry in there that generates the circuit output for breathing, inhalation, exhalation. All that is programmed in the brainstem. Okay, so if somebody has severe damage to the brain, okay, and they're unconscious, all right, and basically almost no activity, they could still, very likely still have activity all the way down the medulla, so they can still breathe. It gets a little dicey in terms of controlling heart rate. But remember what the heart does? Beats by itself, right? So in that case, autonomic control of the heart is just ramping it up or ramping it down. Okay? But breathing, okay, and when we say involuntary breathing, is that circuitry. And when you take a big breath in, you're connecting to that breathing circuitry to drive it consciously. Okay. Particular formation. What do we think about particular formation? Okay. Sorry, losing the back. I should have brought this up sooner. Consciousness. Well, that's a pretty big thing. Be a joke that neuroscientists, when they get older, they stop thinking about like nerve cells and synaptic function. They start thinking about consciousness. I don't know why that's true. It's like, the huge picture, but we understand a bit about consciousness now. But in terms of all the pieces, the visual part, the auditory, how we put it together, think about it, store it, retrieve it to do tasks, we've still got a lot to learn. But it's clear that long reticular formation, remember, runs through most of the brainstem. And there's different parts of it, and they have names. So if you learn neuroanatomy in medical school, you got to learn all these little bits. Okay. Basically, it has this a very strong ascending innervation up in higher part of the brain, mainly up in the cerebrum, right? That helps to basically sync and drive everything so it now is functioning together. And so that's the basic idea around alertness and consciousness. And if there's damage in the reticular formation, people can fall into an irretrievable coma. Okay. 12 cranial nerves. You can learn them if you want. Maybe you went home and tested them. I don't know. There's some interesting things about testing them, but 12 of them. And there are basic functions, just in terms of concepts, okay, basic functions, right? So some of them are just, higher order sensory things like vision and hearing, right? Animals that don't have eyes still can sense touch, right? Primitive organisms can do that, right? Sense pressure. And obviously, if they're marine, then they can even very subtly detect changes in movement. Okay. Now to vision, auditory, olfactory. Obviously, we have an olfactory system. We're going to talk more about this in section 3, but it's not terribly well developed. Certainly not compared to a dog. Okay. A bloodhound that can detect, one molecule out of a billion. All right, enough of that. So, 4 basic divisions of the brain. Okay, so now it's important to know these. All right. Before, when we talked in development, we avoided those encephalon things. So now this is one to know. Telencephalon is the cerebrum and the basal ganglia. So basically, that's everything really above the diencephalon, which is the thalamus and hypothalamus. Remember, 2 things, so there ought to be 2 there. Hypothalamus and thalamus. Okay. There's another bit there too, but we're going to stick with 2. Okay. Midbrain. Okay. That has an encephalon name, but we're just going to stick with midbrain. And then, in this case, in terms of basic divisions of the brain, for gross anatomical, not functional in connection, then we now talk about the hindbrain, pons, medulla, and cerebellum. So the answer to that was actually farther down the study guide, right? Okay. 4 lobes, frontal. Obviously, it's in the front. Parietal, kind of a weird name. That's that association area. Kind of think aside. Right. Occipital in the back. And the back of the brain, when you look at it, even both lobes, it kind of makes a little pointy bit. All right. Occipital. And where's temporal? That's on the sides. But it's not just the sides, right? It also folds down inside there. So, 4 lobes. Obviously, 2 hemispheres. They're connected between this corpus callosum, this connection body. All right? Thalamus is the gateway. essentially for sensory information, right? So all sensory information of all types goes through the thalamus. All right. And then there's that circuitry, that motor circuitry, particularly the basal ganglia. All right. The primary motor cortex is on the front side, so it's actually in the frontal lobe. Somatosensory is actually in the parietal lobe. So even though they're right next to each other and they surround the central sulcus, right? And there's all this interaction, they're designated for two different lobes. Okay. Sounds kind of like an exam question, doesn't it? All right. So, and the other way that I like to think about it is ventral, and the spinal cord is motor. Okay. Somatosensory cortex, parietal lobe, we got that. Visual cortex, we talked about. Auditory, that's on in the superior medial part of the temporal lobe. But if you just think auditory temporal, occipital visual, right? Basal ganglia is composed of the caudate putamen, and this is something to know. Remember we said that one piece of the basal ganglia, remember it's sort of the input part of the basal ganglia, that's the striatum. You might see the word corpus striatum, which adds another part to confuse anybody learning this stuff. So we're just going to keep it simple. This is just known as a striatum, and we're using that word because we'll talk about the circuitry in a second, right? So the caudate putamen, which is, there are really two things. Caudate's got this big tail, and putamen is there, but they're the input part. Then they send their fibers to the inner bits, the little inner wedges, and those are the pale globes, the globus pallidus on the inside, right? Okay, and that encompasses the essential part of the basal ganglia, okay? And again, they're involved with initiation of movement. Their motor patterns come down. In a moment to moment, those change, but as they come down, they all pass through the basal ganglia. It's absolutely requisite, right? All that, if you want to do all this hand waving like I do all the time, that has to be a pattern that's generated going through the basal ganglia, and then acts as a filter to allow that to proceed. And there's adjustments there, and there's big adjustments, of course, in the thalamus. So the output then goes to the thalamus, and then it goes all the way back to motor cortex, okay? And then that's that now a refined pattern with all that other information that's adjusted it, then goes to the output neurons in the motor cortex, and they're the final common pathway, right? So what's the name of the cortical, not on here, is it? The cortical input to the basal ganglia. Well, the input to the basal ganglia is through the caudate putamen, which is the striatum, so it's corticostriatal. Remember, it's always origin, Honolulu to Waianae or something. Okay. The cerebellum computes sensory imbalance feedback, and it can do that independent of the brain, of the cerebrum, let's put it that way. Okay. So that's really spinal to, or vestibular system to the cerebellum. That actually can function and does function completely and independently of the cerebral cortex, or primary motor cortex, or the basal ganglia. There is some input from the cerebellum to the basal ganglia. Okay. But by and large, the cerebellum can function. So what does that mean? You can actually have an animal that has no real function happening temporarily, say in the cerebrum. There's a way to shut that down with anesthetic, but as long as the spinal cord's intact and the cerebellum is intact, you can put that animal on a treadmill and it will walk just fine, because the patterning in terms of walking can be generated very, very crudely, is actually generated at the level of the spinal cord by basic feedback. So the motor patterning for that is actually overlaid on top of that. So the cerebellum functions as this feedback system, but for motor patterning. Limbic system, main thing about emotions, memory, and learning, but particularly with regard to emotional connections to memory and learning, which obviously have aspects that can be independent of emotional learning. So the two key parts to know are the amygdala and the hippocampus. And it already goes on to say the amygdala is involved with linking emotions to memory. An example might be fear of snakes. All right, that may or may not be completely due to it's basically due to some kind of learning that has happened. All right. It could even be verbal initially, right, or emotional in somebody else. You know, like snake comes in the house and the parent jumps up and down, and the kid who normally wouldn't be fearful of it becomes fearful. So it is a learning thing. Hippocampus, processing memory within context. Remember we talked about space. So as you're learning, and this can actually be true for a number of different conditions, even neurological conditions, okay, where even something like addiction very much depends on, which is actually a kind of a learning, right, that happens in a particular context. So it's strongest in that space. So that's why it's really best to take your exam in the same room. Prefrontal cortex, very important for this. And where did that happen? It slept. Searching for source. So we talked about executive function and personality, but one of the key things in terms of memory, we talked about this idea of working memory, okay, as distinct from memory processing, short-term memory processing that has to then get transferred into long-term. The hippocampus is really key for short-term memory. When people have had, and there's occasions where individuals have had either viral inflammation just in the temporal lobe, okay, and they can't really remember anything more than a few seconds, right? You meet them, and then they turn around and come back and say, who are you? So that's really in terms of memory processing, but now you have these memories, you retrieve them, and you got to put them somewhere to do a task. Prefrontal cortex, okay? Questions? So I've added in bits. Do you want to look at anything? Oops. So the other thing that I put online, okay, is a fill-in. Did you see that? So you can go to this, and then what we've done is blank things out. I need to go back and correct it because I actually blanked this out. I don't want you to know the cerebrospinal tract. That's coming from the red nucleus, okay? So anything, if you look back, this box is what we're really saying you should know, okay? So I just wanted to point that out. But that has these boxes blanked out, so then after you go through it, then you go back and test yourself. All right? You can print it out and fill it out if you want. All right, so just to be and firm on these tracks. All right, so you have this sensor out here, as we learn in the third section. They're actually called receptors, sensory receptors, which is confusing. The whole thing's a receptor. So this sensory detector out here, remember it goes through this dorsal part. So again, this is cross section here of the spinal cord. You're looking down on it. This is the back dorsal. So you can see the axon coming in and going into the dorsal column. Column's referring to the fact that it's a dorsal tract. But you just so there's what we call gross anatomy. You just look at the feature as this column of whatever, right, of fibers. So it's requisite going through the dorsal root. So this is the root, okay? And then there's a collection of cells, and there's not one cell there. This is just one example. There's lots of cells here. Therefore, lots of cells. That's why it's a ganglion, okay? The dorsal root ganglion. Goes up, medulla, synapses here. Don't want you to know the gracile nucleus unless you just got to know everything, okay? Then it synapses there and goes across, and this is a little fanciful, but then it does say dorsal here. Goes up to the thalamus, makes a synapse there, synapse number 2, right? And then it goes on up to somatosensory cortex, making a synapse up there. So we count that synapse, okay? That's why there are 3 synapses, from the pressure to the end stage, okay? just so there isn't confusion. Some people look at this and say, well, there's only 2 synapses here, right? Oop, there's one up there. Okay? Spinal thalamic. Okay, so as soon as you hear that, you know that it started in the spinal cord and it's gone all the way to the thalamus, right? So you got to think to yourself, what happened? So that's the one where now you got pain and temperature, and you do get a little crude touch, but we just left that off. Going again through dorsal ganglia, coming in, and in a particular patch here in the dorsal part of the gray matter horn, okay, makes a synapse, crosses, goes through this patchy bit of track that's underneath the ventral horn. So it's not strictly true in terms of sensation being dorsal or ventral. In this case, a very important sensation. Already crosses, goes through this now spinothalamic track all the way up to the thalamus, right? So, and you can track it all the way. It stays on this side. Another synapse going all the way up 3. Okay? And then motor, as I mentioned. There's all this stuff that happens. Then the pattern comes down through lots of fibers. This is only just showing one fiber, right? Comes down here into the bottom of the thalamus, crosses over, and that funny name for that is decussation, okay? And they make little pyramids, so they call them pyramidal decussations. In fact, if you look at this part of the brainstem and you look there, they actually kind of interdigitate. You can see it grossly on the brainstem. You can say those are exactly the crossing pyramidal tracks. Okay, you can actually see these little bits. So you know where that is exactly. Very bottom of the medulla, then comes down through the lateral cortical spinal tract. Okay. To the motor neurons, where are they? They're in the ventral horn, right? So these fibers go out the ventral route to the muscle, to particular muscle fibers. Okay? Now, one last little bit that may come up, all right, is this primary pathway, since it goes through these, quote, pyramids, it's called the pyramidal tract as well. In fact, it's described as the pyramidal tract until it gets really down the spinal cord, where now it's called the cortical spinal tract. But if you just look here, this is not necessarily referred to as the cortical spinal. It's the pyramidal tract, which is the primary motor pathway. So any other track outside of that is called extrapyramidal. Any other motor circuitry is called extrapyramidal. And what's the biggie that we refer to as extrapyramidal? That's the basal ganglia, right? So we talk about extrapyramidal system, usually you're talking about first on the list. Not only on the list, but first on the list is the basal ganglia. Okay? 2 synapses. And they're the cranial nerves. Questions about this? I'll bring up the other neuroanatomy PowerPoint, but I want to leave this available. Good with this? Okay. Just got to sit down and remember this stuff. Okay. Anatomy is not conceptually difficult at all. It's just, names, places. All right. So this is just a little side. But I can guarantee you that this brain has the P on it. Okay? Because it looks kind of like a package, doesn't it? Where's the front? It's oriented the usual way. Sometimes you're really not clear. You say, well, where's the cerebellum? Oh, there it is, cerebellum in the back. There's the front. And then you see here, it's kind of hard to see the temporal lobe, isn't it? Because it's packaged in that pia mater. If you peel that off, okay, and it sticks and it goes inside, you have to be very careful. It goes inside all the crevices, the sulci, right? So you have to peel it out carefully. Okay. You know how to orient yourself. Basic divisions. Okay, we've gone through that. The lobes. So only 4 of them, right? Not so hard, but frontal's the easy one. Okay. And then something I alluded to at the corpus callosum. Remember we talked about these fiber tracks There's these short ones that run between the gyri, you know, either neighbors or a few neighbors over in the cerebral cortex. And then there's the ones that communicate front to back, all right? And they lay over the top of the ones that run side to side. And that's what we see here. So these are underneath all those front to back ones. And these are basically point-to-point connections, right? So that you have bilateral communication. Now, later it may come up that there may be some little discussion about asymmetry in the brain, but we're not going to really get into that in any big way. So obviously there are differences. There is a dominant hemisphere. It's usually by handedness, but not always. All right. So if you know you're right-handed, your left hemisphere is much more dominant. All right. But be that as it may, even if that's true, where you've got more dominant activity over here, it's communicated to the other side. Thalamus, remember it has connections from the cortex back to the thalamus. All right? But that's basically like a feedback adjustment system, right? So that's a little higher order for complicated thing, right? And then we talked about, we'll get to it in a minute, talked about where the different bits are, the motor and the somatosensory, the basal ganglia, and the cerebellum here. So this is important to know. Again, we've gone through this. I do this descriptively, but you know that patterning comes out of the motor. Basically, you need to know motor, but it's really a combination of motor and associated premotor areas. Goes through the caudate putamen, so that's cortical striatal, right? So you could write that in here, all right? Then it goes globus pallidus, thalamus, all the way back up. So that's that important processing loop, all right? And that's extrapyramidal, right? Because the primary output goes through the pyramid, therefore is pyramidal. In fact, down here at the bottom, I don't know if you can see that in the back. If you look at your PowerPoint, that distinction is here. Okay? Cerebellum, this is the pathway here. So the one addition to add, I mean, there's obviously a cortex to the cerebellum, but the output is through these deep cerebellar nuclei. Okay? Review of that. Okay. The key things here: amygdala and hippocampus. Don't worry about Phineas Gage. Tell your parents about that. Hearing is going through the medial geniculate. Vision going through the lateral geniculate. Okay. Any questions? I'm going to go to the study guide on the developmental neuroscience. Yes.
I just downloaded that one from Lena and it's on his last 3 slides on it.
Okay, I'll double check that. Should. Maybe I left off. This is the power. Well, I'm looking myself. Doesn't make sense because this is the power. This is the PDF that was mounted. I'll double check that. Okay, sorry if that's true. Does anybody else see that? These are on the. This is the PDF loader on the level 1. I'll double check that. Sorry. So I want to go through this really quickly so that we have the time, but I don't want to give any short shrift to this. So, no other question about neuroanatomy? It's straightforward. Try to keep it with names that sound like what they are, like, you know, brainstem or hindbrain. Okay. Instead of all the funny names like, you know, encephalon this and encephalon that. Except for telencephalon, diencephalon, but then should know what those are. Okay, let's get on to the development. Going backwards, aren't we? So I have these in 4 sections, and again, we can bring up PowerPoints if there's something there that you want to see. I thought I'd just go through this, and in the interest of time, not bring up the PowerPoints unless you're really dying to say, okay, that doesn't make sense. Can we look at the original? Okay. So, in terms of this background with regard to the development of the nervous system from an evolutionary point of view, okay? Try to collapse it all down this. And this was that one of the last slides. Well, it was the slides after the evolutionary bit, okay, in the first session. Okay, so this is all there. So this is just condensed that down. The idea that in very primitive organisms, the metazoan, the porphyra, was basically the sponges. No nervous system, but they have these prototypic or protosynaptic proteins, right? They have proteins that are basically homologs. And again, that all evolved from basically moving stuff around by vesicles fusing to membranes to deliver something here and there within the cell. That's now adapted, right, to make a synapse. So those are all there. Okay, first are the diffuse nerve nets. We see that with an idaria, like jellies, right? Okay, appeared. They have bona fide synapses, but they're more often than not, they're bidirectional. That means if you poke on one side, you know, there's really no orientation. They are symmetrical. They're radially symmetrical. But basically, information just kind of wings around inside there. So they get stimulation really that evokes activity across the system in any direction. We see that they have limited capacity for locomotion. You know, they kind of do this kind of thing, although in some regards, you know, it seems quite elegant, but they're really not, it's not directed, it's not doing anything that's really task-oriented. That really changes when we start to get to the next stage, when you get centralization, connection with nerve cords. See, that's worms and mollusks, but the most remarkable, of course, are the cephalopods. So now they can do very sophisticated locomotion, which is what we saw with the octopi. And then that continues again, so that we now go from, you know, even though we have polarization, right, centralization, and then you're sending out information, all right, sending out signals through nerve cords. Now we go to bilateral symmetry, progressively more complex plans, organized, centralized, segmented output, segregation of function into voluntary and involuntary. So that was something that was obviously lurking under what we talked about with the neuroanatomy, right? So moving your muscles is voluntary, you know, controlling your heart rate by and large is involuntary. Even if you can get to a Zen state, you can lower your heart rate. That's very unusual. By and large, everything is, in terms of basic functions, is involuntary. Okay, so that evolved later, so that you've segregated function, you've segregated activity. So you have a particular sensation or particular thing that organism is going to do. So you make a specialized system for that. And a big part of that is, in terms of locomotion, you know, getting around and finding things and eating them. Okay, those are pattern generators. All right. So those are really the basic things from this. I understand that this is, you know, related to development of the nervous system, but it's a different type of development of the nervous system. This is evolution, and now we're looking at the next bit. You know, how does it actually develop? So, and this is really looking at more complex organisms, right? So we're really talking, you know, with chordates, with vertebrates. They generate a neural plate, okay, that neural tissue derives all from the ectoderm, okay? The ectoderm sits over the top of the mesoderm, and the mesoderm has to it actually sends signals, not the morphogens, but there's inducing signals going unnamed, all right, that induce the ectoderm, let's just say part of the ectoderm actually, key part, the central part of it, to become neural. So then that part forms a plate, and that plate folds up into the neural tube, right? Neural tube differentiates into 3 vesicles, and here they have, again, these encephalon names with just forebrain, midbrain, and hindbrain. But remember the 2 brainstem bits from the hindbrain, medulla and pons, together with the midbrain, make the whole of the brainstem. Okay? 3 major strategies. These are basic strategies to know, all right, in terms of why identical cells, as they're generated, then take on different features. This is the whole idea, not just of differentiation, but you can get, you know, patterning for differentiation of types of lineages of cells that eventually have to segregate, as we said, to have different functions, okay? So you could have mRNA localizing differently. I wrote here unequal division. What I really meant there, I look back at this, is that you have unequal distribution of the mRNA during division. That doesn't mean you have a little cell and a big cell during division. You have 2 cells, and this one has, you know, a little bit different amount of RNA. Okay? And then, of course, transport. So the RNA may be moved to a different part of the cell. Cell-cell contact, cell-cell receptors, intracellular cascades. The key thing here is that this is leading to changes in terms of expression. So with the mRNA, you know, you have different mRNAs over here. That's going to be a different set of proteins in this cell, right? And then now you have cascades that will, say over here, this cell gets a signal that's different than this cell, even though they start out kind of similar. The signal's different here, it's going to drive different gene expression. So that's lurking underneath everything, right? And then the diffusible signals, concentration gradients, that's a big part of why different cells will have differential expression. And remember, we talked about the morphogens. Some, you know, cell close to the source of the gradient would get all of the, you know, the diffusible substance, you know, that morphogen, okay, that signal occupying all of its receptors, get a big signal that drives the transcription of a whole bunch of genes, and then the cells successfully farther away get a, you know, they get less signal, less stimulation intracellularly, drive the system less, fewer genes get induced. And then there's this example about delta, right? So there's been some question about this in terms like, okay, what does this mean? So, actually, sorry, I can't find my mouse, all right. Let me just bring that one out really quickly. Or not. Where are you? I think it's this one. Yes, so what do we really learn from this? So there is a bit that I decided not to have you worry about, okay, and that's this. So when there are cells that are now committed to become neuroblasts, okay, there's a lineage there, and they actually have this arrangement, and there's a key cell here that we didn't really go into, and I don't want you to remember, this ganglion mother cells. But the thing is, you now have a stem cell organization where this is spitting out the neuroblasts that then differentiate to neurons. So they're generating them. So when the neuroblasts are generated then from this source, that's why they inhibit the surrounding epidermis, okay? So that's the potential confusion. It's like, okay, well, if the neurons are inhibiting the epidermis, why aren't they inhibiting each other, right? But they're being generated from a unique source. So that's the difference there, okay? So when that happens then, all right, and you get neuroblasts, and I gotta find my mouse, all right, then we have this delta that's on the surface of the neuroblast. It binds to this notch, okay, so the cell surface delta, so it's now, you know, sticking out, and it's interacting neuron to epidermal cell, delta to the notch, and then the notch drives the neuronal repressor genes to keep those epidermal cells from becoming neurons. Okay, and that way you get get a nice patch of neurons sitting in a sea of epidermal cells. So that's one example. The other thing, though, in terms of what's going on in vertebrates, in terms of the generation of these cells, okay, now in the nervous system, they're being generated in this ventricular zone, this bottom zone, the more inner zone compared to, say, the outer cortical areas, okay? So there's those rounds of divisions. They start first vertical cleavage, so they just make a bunch of more neuroblasts, and then that converts to horizontal cleavage, and one of those daughter cells, it goes up and differentiates, and it'll go out and become, you know, a differentiated neuron in the neuroblast, and in the cortex. And in the case of the cortex, those pyramidal cells, those really important pyramidal cells that make up, you know, they get all the information that's computed in that cortex and send out the key information. They travel up those radioglial cells. Okay. So the key thing in neurogenesis is the early stage of cleavage, that is division. So you cleave locally, and then you just keep making more and more neuroblasts, and that converts later to where you spit off one that will go out to differentiate. Okay. And this happens at a huge rate in humans. You know, millions of cells, hundreds of millions of cells at one point. All right. Real quick, we'll go, because I want to wrap up here, so you have plenty of questions to ask on your own or as a group. Axon guidance, it's the growth cone. So know the growth cone. Okay. What do you need to know about the growth cone? You have to have a picture here, wouldn't it? I don't know where it is. Why can't I find my stuff? Okay. So key things are what it does. So it locomotes, it moves. That does. There is a certain amount of tension, but that is, as it's moving, it's driving addition of membrane to make the axon grow, right? So it's finding its target, but it's also extending. So it's moving toward that thing, but as it's doing, it's making more. The system behind the growth cone is making more axon. So it's attracted or repulsed via molecular cues. They can be diffusible, contact mediated, and so you should know these things. The growth cone. Peripheral domain, right? In the peripheral domain, we have these finger-like things. Those are the filopodia, single filopodium, lamellopodiums. Okay, that's this bit in between. That's this whole thing here in between. So distinguishing between this finger and this. But you can say this is all basically a veil of lamella. So it's not unlike what you see with other cells that migrate. They put out this big sort of sheet. Now in the growth cone, it's adapted to that to have little fingers, with receptors on them and things. And then there's a high density of actin. So when you think about the periphery of the growth cone, you think about actin, right? Central one, okay, what are these things? These are microtubules. They're stable microtubules, particularly in the axon. And again, we really aren't going to talk about axonal transport here, but they have to be stable because they're bringing things. They're more dynamic than out here, but they extend all the way out to the base of the filopodia. Excuse me. And then there's this actin network here, basically kind of a rim. And what is it? It's really an anchor then for the heavy F actin that's out here in the really active part of the growth cone, because remember it does this. So when it's doing this, it needs to have basically an anchor cytoskeleton for that. Okay? And then how does it move? So you should know these things. How does it move? Actin treadmilling. Now I know this can be a little bit of a confusion. So let me just go over this quickly, because sometimes people say, well, it seems to be going the wrong way. I don't get it. Okay? So imagine this is F actin that's projecting out into into the growth cone. It's in part of that network underneath. So it's linked through these proteins, all right? Lots of names, don't need to know them, but they do interact with the thing you do need to know, which is that integrin. And the integrin has two sides to it. So this is anchored to the F actin, all right, through these guys, and then it interacts with bits in the extracellular matrix and primary laminin. That's what the LN is, okay? It can interact with a collagen and some fibronectin and other stuff, but the one I want you to know is this laminin, because it's key, all right? So that forms a stable, tight interaction, moment to moment, okay? But it can break apart, so it is also potentially dynamic. But moment to moment, when that growth cone's sitting there, for that one moment in time, this is all rigidly stuck together, right? So then this thing is treadmilling. So which way is it going? Okay, it's adding to the front, taking off the back. So what it's doing is it's shifting this way, all right? Well, where's this going? It's going that way. So that's why the directionality is forward. So it's generating force that's pushing the whole thing this way. Really. That's what, if you look at it, that's what it's doing, right? So that treadmilling is key. And then in terms of movement of membrane, all right, there's addition of membrane that has to happen as it's moving forward to grow. Okay? So we talk about actin treadmilling that generates retrograde F actin flow. Where's my mouse? Who knows where my mouse is? There it is. And And that's through, this is split, sorry, between integrated receptors, the F-actin associated proteins on the inside, lamina on the extracellular side. So membrane in addition to the front. And so what the attraction cues and the repulsion cues do is to foster dynamics one way or the other, right? Traction to help drive it to locomote forward or to disconnect for repulsion. So by and large, these are inhibiting these interactions, and what we're saying primarily is the interaction between the F-actin, the integrin, and the laminin. Okay. So you see that so with repulsion cues, they dissociate. I mean, not completely, but the dynamics of that are such that repulsion cues will slow down. So if you have a condition where repulsion, a very strong repulsion, what you'll actually see is the growth cone will just stop and do that. Okay. If it's like a really high concentration everywhere. If you have a gradient, okay, it'll more likely be able to pull and turn. So repulsion can turn, but if it gets really close to the source, it'll literally stop the growth cone, and that's because it's disrupting all those, those locomotive actions. Okay. Netrins. So you should know basically these 4 types of molecular cues: the netrin slits, the semaphorins, and ephrins. The netrins, they signal through the DCC receptors for attraction, the N5 for repulsion. Slits are a repulsive signal. This was a mistake on what I mounted. The correct version of this guide is on Lalima now. So these are repulsive signals. They work through this robo, and remember that example. Okay, so semaphorins, they have these receptors, ephrins have those receptors. But then remember they have the example of nephrin. In the spinal cord, it'll be in the midline, and it'll attract fibers to the midline. And as they cross over, then there's a switch in terms of expression. So the growth cones are expressing DCC, attraction. They switch to express robo, because there's also slit being made there. So once they get past, you can actually now have a repulsive signal that helps to move it away. So it's really, it's guidance, right? But it's saying, okay, you've come here now, go away. So even though you think of it as repulsive, it's repulsing it to make it go farther. It's basically guiding it to another place. Okay? Important to know that. Synapse formation, okay, we are getting a little behind her. So axonal guidance, key things there. So that's obviously a big part of synaptic formation, but that's covered in the other bit. So we talk about target recognition, selection, and synaptogenesis. So key things is that we have those laminins, right? Cell adhesion molecules, okay? But the thing you need to know is what they do. Is that when the growth cone comes to its target, and that's where the synapse is going to be, particularly cell adhesion molecules, but also the laminin, okay? It converts necessarily from a substrate for, on which locomotion happens with the growth cone. It also becomes the cell adhesion molecules, and it sticks that bouton onto the synaptic site, okay? So adhering that. And then there are neurotrophins of all different kinds, with the classic one, nerve growth factor promotes survival activity, okay? And so as development goes on, there's a lot of growth, a lot of synapses are made, and then ultimately there's a limited amount of neurotrophin, and then those synapses basically compete for that neurotrophin. It's to some degree random, but not always. They can't all get it, they don't all survive, so then you get pruning back to just the essential ones. So the system adjusts to have just enough neurotrophin for those synapses to form to whittle down to the key ones that matter, right? So you have pruning of, sculpting or pruning of connections, and that goes all the way back then. So once they're pruned, they're not getting neurotrophins all the way back to their cell body, because they take them up and send them back to their cell body to say, okay, good. So now if they're not there, not only the synapses, but the whole axon and the whole cell dies, okay? And the example of the neuromuscular junction, okay, where it had binds to the beta laminin, then there's this agrin that's secreted, binds to this this kinase, this agrin receptor, that activates rapsin, and that drives localization. So now you make a synapse, it gets the adhesion, neurotrophins help to say, okay, you need to stay there, all right, you're good. And then now through the signaling, it now brings all the signaling receptors there. And remember, activity locally matters for solidifying that synapse. So now you have a lot of receptors there, they get activated, they send signals back And the key thing they do is they send signals back that stimulate calcium entry presynaptically. So that's going to cause potentially some neurotransmitter release, but it's done in a more, it's not linked to a signal like an axe potential. It's just sort of a long change, a long rise in calcium in that terminal, and that helps to activate things that set it in place. Okay. And then there's this neuregulin that regulates the local acetylcholine expression. That is the number receptors. All right. Okay, and then lastly, we have the fact that, again, you have this pruning, an example in neuromuscular junction, or the neuromuscular innervation, that basically it's all over the place, and then it gets pruned back such that, so we call that polyinnervation. Eventually it goes to, one axon to a synaptic site per muscle fiber, okay? And then the higher order thing in the brain, that when things are together now at the level of synapses in the brain, that when they are firing in conjunction, that helps to solidify each other. So they talk about this idea of fire together, wire together. Okay. I want to make sure that we got through all this. I didn't see hands going up, but questions? Yes?
You had a section on there for, I think, the summary of one, and then might be this section here, and it said rearrangement and plasticity, and it had two things on it I was kind of confused on. There's experience within it, modulatory influences.
So the question was, there was that, after, at the end of this, there was that thing about, the first was this fire together, wire together, and then there was that other part about modulatory influences. That's interesting, but we're stopping here. This is Hebb's rule. Yeah, the experience-dependent stuff. Yeah, we're not gonna work. Some of that was in that other lecture you had in section 1.
Okay.
And I don't want to overlap too much. So yes, experience helps to drive this kind of thing. Just that one. This is what we have here. So I'll stick around.